Hi, everybody. This is Dr. Bob Roundtree, and I'm really excited to present to you what is really the first and most foundational lecture of this advanced practice module on immune disorders. So this particular lecture is called Underlying Concepts of Immune Dysfunction and Inflammation. And this will be followed with the live on-stream part of this presentation. So my disclosures are here on this slide. I do consult for a couple of companies in the area. And here's where we always start in functional medicine, with the matrix. And this particular module is focused on defense and repair. And defense and repair really encompasses immune function, the inflammatory process, how the body deals with infection, the whole concept of the microbiome, how do all these things interact to affect health, wellness, and disease. Now, these first two lectures, part one and part two, are really a deep focus on the antecedents, triggers, and mediators of inflammation and immune disorder. So we're going to go down the rabbit hole into molecular medicine, the whole concept of what's going on a cellular level that creates the phenomenon that we see as clinical disease. I want to start by talking about healthy immunity rather than dysfunction. And there are a few things that we know about healthy immunity. Number one, the goal of a healthy immune system is to be detective and defensive. And there are two aspects to this detective phenomenon. Number one is identifying potentially threatening molecular structures. And it's really important to emphasize that word, potentially threatening. The way this is described in the immunology literature is stranger signals, something that signifies a difference from self. Strangers are not self. And where do we find these stranger signals? In microbes and food. But our immune system doesn't just react to stranger signals. It notices them. It's aware of them and is primed and prepared to react. But what really induces the immune system to respond is host-derived danger signals. These are molecular structures that are released by distressed or damaged tissues. And so what that basically does is it allows the immune system to mount a response that's appropriate to the level of threat. If there's a stranger but no threat, then the immune system simply goes on alert without reacting, and that's exactly what you want. The number two major function of healthy immunity is to be internally regulated, and the word of the day is homeodynamic. What that means is the immune system doesn't just react and that's the end of the story. Immune responses are very tightly controlled and they're actively resolved. Anytime inflammation is induced in the body, it's also turned off from the very beginning. They're very tightly controlled, multiple genomic and enzymatic mechanisms that do this. The third aspect of healthy immunity is that it's restorative. It repairs damage that results from trauma. So if you have a cut or a sprain, the immune system comes into play, produces inflammation, but at the same time it produces that inflammation, it also starts the repair process of any kind of adversarial encounter. And then finally, the immune system is tolerant. It's actively unresponsive. The word used in the literature is that it's anergic instead of allergic. It's anergic. And anergic means that the immune system is actively recognizing these stranger molecules, but it's not necessarily doing anything about it. There's a lot going on in the cell as a result of this recognition. What is it recognizing? It's recognizing self-antigens, And it's recognizing specifically self-associated molecular patterns, sometimes called SAMPs. It's also recognizing microbial antigens from commensal bacteria that normally populate the gut or the skin or the respiratory mucosa. These are all innocuous antigens, and the immune system should not react to them. But the lack of reaction doesn't mean that the immune system isn't seeing them. It's simply seeing them and then making a, quote, decision not to do anything about it. That's tolerance. So tolerance is a really, really key phenomenon to understanding everything we're going to talk about throughout this whole module. Think about it. In the course of a lifetime, we literally ingest tons of food. Tons of food go through the GI tract. And we simply can't have an immune system that's reacting to all that food, or we'd be in trouble. Now, clearly that does happen in some people. They do start reacting to lots of foods, and that can cause serious ramifications for health. So you put all these together: detective, defensive, internally regulated, restorative, and tolerant. And what do you get? You get dirt. And that's no accident that put this mnemonic together that way, because one of the problems in our society is that we are a bit too clean, which means our immune systems don't get properly programmed and aren't as fully charged as they have been in the past. So how does this whole system work? How does the immune system work? I'm going to walk you through a timeline, which is really a microcosm of the timeline of what happens with disease. So there's the macrocosm of the timeline, where a person gets an infection, they take antibiotics, they get dysbiosis, that sets into motion all kinds of other problems in the gut. Well, that's the macro version of the timeline. I'm going to talk about what's going on a physiologic level with the timeline. So it's a slightly different reframing of what that timeline is about. What is it that keeps pathogens at bay? Well, number one is physical barriers. And those barriers are actually the tissue itself, and then the surrounding biofilms and other layers like saliva and mucus on top of those physical barriers. So that's the first thing that keeps pathogens out. Number two, in the respiratory mucosa, you have cilia that are constantly moving, and that basically brushes pathogens up out of the lungs into the mouth where they can be expelled. You have an acidic pH, and you have enzymes like lysozyme, which is found in tears and mucus. So here is a picture of that process. The big circle is the physical barriers, which we might think are inert, but in a minute I'm going to show you something that I think is mind-blowing. But that's a traditional way of thinking about the physical barriers. Is they're an inert obstacle, impediment to pathogen entry. If the pathogen, the virus or the bacteria, gets past that physical barrier, the first thing it's subject to is a non-specific control. That's the innate immune response. The innate immune response goes from non-specific to more broadly specific. And then over time, in this timeline that we're talking about, we eventually get to a highly specific control, which involves antibodies and T cells, which includes T helper cells and cytotoxic cells. So we go from completely non-specific, like a barrier, to slightly specific, broadly specific, and highly specific. And all these things take place over the course of about a week to 10 days, which is exactly why with a typical respiratory tract infection, it takes about that long to get it under control. Now, this is a fairly new understanding that has really become kind of revolutionary in terms of what is going on with the innate immune response. It turns out that epithelial cells and barrier tissues retain memories of previous immune events. Wow, think about that. Think about it. Let that sink in. That barrier tissues can recall environmental exposures. exposure to microbes, antigens, and other noxious stimuli. And this barrier memory basically allows us to respond more quickly when exposed to similar kind of pathogens, to similar kinds of viruses. And if this barrier memory is not there, then we have increased susceptibility to infection. Or if that memory retrieval is excessive, if the barriers are subject to exposure over and over and over again, then we end up with chronic inflammation. Or if there's insufficient barrier memory and perhaps excessive inflammation at the same time, we can get malignancy. So this is clearly a whole new area of studying what's going on with inflammatory memory and how do we make it work for us instead of against us. It's no accident that epithelial lymphoid tissues are on the front lines of defense. So right underneath the barriers, what do we have? We have these collections of lymphoid tissues in the respiratory tract. We have the nasal associated lymphoid tissues, the NALT, the bronchial associated lymphoid tissue, the BALT. In the skin, we have specialized lymphoid cells. This would be called the SALT. In the GI tract, the gastrointestinal associated lymphoid tissue, the GALT. And then in the urogenital mucosa, we have the VALT. So this is the front line of defense. This is where most of our immune system is located, is right there where it's exposed to the outside world, which makes a lot of sense. Now, obviously, most of the immune system is there at the GALT, right? Because that's where the major exposure is to these strangers or potentially dangerous strangers in the GI tract. So obviously, if we want a healthy immune system, we've got to have a healthy GI tract. So I mentioned we get past the barriers, then we start moving into a more broadly specific and induced response. This is all part of the innate immune system. This occurs within the first few days of exposure. And the first guys that are on the scene, I probably shouldn't be gender specific on this. They're not guys. The first players that are on the scene, if I'm going to be correct here, would be the neutrophils. They're the early responders. Okay, quickly followed by monocytes, which are transformed into macrophages. Both of these are phagocytic cells. Then we have antigen-presenting cells, which do include macrophages, but more specifically dendritic cells. And we'll talk about those in some detail. And cytotoxic cells, natural killer cells, which move into the site of injury and start degranulating. They release toxic chemicals that help damage infected cells, especially viral infected cells. So here is a picture of a neutrophil. As I said, one of the first players on the scene engulfing anthrax bacteria. And you see it as it's a major capacity to expand and move and engulf that tissue. And the question is, well, how does the neutrophil know that this is something that it should eat? And that's going to be the subject of a much more detailed part of this lecture. We're going to come back to that, the recognition patterns that lead to this whole process developing. Another part of this broadly specific induced process is the release of pro-inflammatory cytokines. In the case of viruses, it's especially the type 1 and type 3 interferons that are playing a role. And from time to time, I'll be making mention of what's going on with the current pandemic, which is that we're learning a lot about which parts of the immune system are particularly important for fighting off viruses like the SARS-CoV-2 virus. And it turns out interferons are extremely important for fighting off SARS-CoV-2. And when the interferons are deficient, that may lead to a more serious inflammatory response in the future. We also activation of complement, big proteins that are floating around in the bloodstream and circulation that get activated when they come in contact with antigens, break apart and form a cascade. We have antimicrobial peptides like alpha and beta defensins and cathelicidin. We have the respiratory oxidative burst with reactive oxygen and nitrogen species that are produced by very specific enzymes in immune cells and other innate cells. Basically, this respiratory burst is like pouring bleach on the foreign invader. It's as if the infidels were heading towards the walls of the castle, and you have the first line of defense, the soldiers at the top of the wall that are pouring bleach, hot toxic chemicals onto the invader to kill them off. Here's a picture of that happening. An injury to tissue activates white blood cells. We see macrophages and neutrophils producing free radicals, nitric oxide, superoxide, hydroxyl radical. All of these things are helpful in that they kill off microbes, but at the same time, they can damage surrounding tissue. And that leads to kind of one of the first concepts about how to keep the immune system healthy, which is eating a diet rich in antioxidants, a diet that keeps levels of glutathione and vitamin E and vitamin C up. What's the advantage of that is that these free radicals can damage tissue, and we don't want collateral damage going on like that. So protecting tissue with a lot of antioxidants, especially the ones found in plants, can keep this collateral damage from occurring and strengthen the immune response at the same time. Once we've gotten past the defenses of the innate immune system, that moves us into the acquired immune system, which is mediated through lymphocytes specifically. And that typically takes 4 to 5 days to respond. The two major categories of the acquired immune response: the cell-mediated response and the humoral response. The cell-mediated response involves T helper cells, which have a cell surface marker called the cluster of differentiation 4, CD4, and cytotoxic T cells, the CD8, cluster of differentiation 8. Now, these cytotoxic cells are a lot like natural killer cells. The difference is that natural killer cells, which are part of the innate immune system, are already in circulation. They're already ready to respond to certain molecular structures, whereas cytotoxic T cells need to be woken up. In fact, that's true for all of the cells of the acquired immune system, is they need to be awakened. For example, B lymphocytes. There's some in circulation. We have what's called memory B cells that are in circulation that are capable of recognizing certain antigens, but they're there in very, very small quantities. So when a bacteria or a virus invades the system, or if there is a vaccination process going on, that wakens up the lymphocytes, they start proliferating. And it's that proliferation process that leads to their conversion into plasma cells and the ability to make antibodies. So here's an overview of the innate versus the adaptive immune system. You can see that there are many players in the innate immune system, not just immune cells, but also epithelial cells. And there's monocytes and macrophages, dendritic cells, neutrophils, eosinophils, basophils, mast cells. We're going to talk about all of these to some extent. Then there's cells that are kind of in an overlapping category, like natural killer cells or natural killer T cells, gamma delta T cells, and innate lymphoid cells. They come from a slightly different lineage than other innate immune cells. In that sense, they're similar to adaptive immune cells, which would be the T helper cells I talked about, the cytotoxic T cells, the B cells, and then a unique group of cells called T regulatory cells, which we'll go into in some detail. Really what this lecture is all about is identifying the players. so that you can understand what all these different cells are doing in the immune response. What role are they playing? So here's the big distinction between the innate and adaptive immune systems. On the left, we see the innate immune cells, which involve neutrophils, monocytes, or macrophages, natural killer cells, and complement. And they are primarily activated, they're primarily working through phagocytosis or oxidative bursts. In certain cells, that leads to antigen presentation and cytokine production. That antigen presentation then activates T cells and B cells, leading to proliferation and antibodies. The cytokine production, the pro-inflammatory cytokine production, in turn activates proliferation and induces antibody production. So we start with the innate immune response, and the innate immune response then gives way, segues into the adaptive immune response. You can't really have the adaptive immune response without the innate immune response kicking in first. So our original understanding of how the immune system worked really came out of an initial debate, which was a belief that there were two major arms of the immune system, a cell-mediated arm that somehow involved the cellular response. The different types of lymphocytes weren't really understood, but that there were cells involved, and there was some other versus some other mysterious substance in the blood, which was called humoral immunity. It was a humoral mediated response. So that was originally how they thought about the immune system. It was only much later that there was a realization that there was an innate immune system that didn't require prior exposure, and an acquired immune response that required exposure through, say, vaccination. So what we know now is that the innate immune system is hardwired to recognize certain molecular patterns. And I call these generic patterns. So there's 1,000 or so patterns that are programmed into the DNA of innate immune cells, including epithelial cells. So they simply recognize the pattern and they respond. And how do they respond? They respond with a controlled inflammatory cascade. Now, the IFMCP course includes a lecture that I give on inflammation, and I do suggest that you go back and listen to that lecture again as a review. I will be touching on it again in part 2 of this presentation, but just so that you're fully prepared to understand what's going on there. So part of the way that the innate immune system repels invasion by pathogens, dangerous strangers, is by fully activating that inflammatory cascade. Something that I want you to get is that the innate immune system shares very similar genes by all classes of life, really. Plants, insects, and animals all have very similar receptors, all have similar genetics. The big difference between plants and animals is that plants don't have mobile defender cells. They don't have circulating immune cells, but they have many of the same receptors that can recognize the patterns of dangerous strangers and respond appropriately. I find that somewhat mind-boggling. Now, when the innate immune system was first being mapped out and understood, it was assumed that it was very primitive, very rudimentary. This is something that mostly you're going to find in jellyfish, right? And so it was really downplayed as being very, very primitive. But now we understand that the innate immune response really acts as the gatekeeper for everything else that's going on in the body. The dendritic cells, the antigen-presenting cells in particular, are the main determinants, the mentors that are going to set a direction for how the rest of the immune system responds. That is really a really profound concept because it's the innate immune system that's interfacing with the outside world. The innate immune system is interfacing with the foods that we eat and the healthy and unhealthy microbes that line our skin and our gut and our respiratory tract. So if the innate immune system is happy, then everything goes according to plan. If the innate immune system is under-functioning or over-functioning, that's where we get into a lot of problems. What's the role of the innate immune system? To identify and remove foreign substances. Okay? Garbage disposal. Garbage disposal, especially when it comes to macrophages. The innate immune system is also about recruitment. It's your local military recruitment branch. When there is an injury, the immune cells will start releasing chemokines and other soluble messengers that will go out into the bloodstream and summon other immune cells to the site. The innate immune system also involves the complement cascade, which are these big circulating proteins made in the liver in circulation all the time. These proteins come in contact with foreign molecular structures, strangers, and specifically dangerous strangers. The complement proteins will break up and they form what's called split products, sometimes called anaphylatoxins. And these split products are highly, highly inflammatory. They activate immune cells like mast cells and macrophages. And then finally, something that I've just mentioned is the innate immune system is involved in presentation of antigens and priming signals to the adaptive immune cell. And that's the system by which the innate immune system regulates the adaptive immune system. We're going to go into that in some detail because that interface between innate and adaptive is really, really critical. And I can tell you that modern pharmaceuticals are really looking at that interface in some detail for controlling cancer or autoimmune diseases. So one way to think about innate immune cells is that they're sentinels. And as I was preparing for this talk, I was looking at my cat who was sitting on a counter doing what cats do, which is they sit there and they kind of silently scan the environment. They look for something interesting like a bird or a mouse. Okay, they're sentinels. My cat is a sentinel. And what are these sentinels? Well, they're phagocytic cells, which I've mentioned, neutrophils and monocytes. The difference between monocytes and macrophages is that monocytes are the main cells in circulation. And then when they get transformed into more active form macrophages, they're found more in the organs. We have mast cells, which are found in tissue, in the skin, in the respiratory mucosa. And we have circulating eosinophils and basophils. We have a really important group that Dr. Polly Matzinger has termed professional major histocompatibility complex type 2 antigen presenting cells, professional antigen presenting cells. These are cells who have a lot of their machinery devoted to presenting antigens to naive T cells and converting those naive T cells into a differentiated cell type. So that includes mostly dendritic cells. They have the broadest range. Also monocytes, macrophages, melanocytes. Yes, melanocytes in the skin can present antigens. B lymphocytes can present antigens. They're not technically part of the innate immune system, but in this case, they work a lot like innate immune cells. And then finally, the natural killer cells, which are a subset of innate lymphoid cells that I mentioned earlier. They're cytotoxic cells. Very key concept. Most innate immune cells are initially in a dormant or resting state. Like I said, very similar to my cat, just kind of hanging out, looking around, waiting for some action. Polly Matzinger says these cells are sleeping. She says they're sleeping. They're floating around the bloodstream. They don't seem to be doing much. Their cellular machinery is not activated. The DNA is not turned on. and they require several signals, typical signal being a cytokine, which would be more like a danger signal, to become fully activated. Again, we keep coming back to this concept of the dangerous stranger. That's what's required to really get the immune system going. Now, I mentioned that barrier tissues have a memory. This is true for all innate immune cells. So, yes, the distinction between innate cells and acquired cells is that innate cells are hardwired to recognize certain molecular patterns. But guess what? They can respond to those patterns in a quicker and quicker manner over time. And this is really what the BCG vaccine is about, to enhance the person's ability to respond to tuberculosis. It's a training of the innate immune cells. So they get exposed to a particular molecular structure, and that allows them to respond more rapidly after repeat exposure to an antigen. That's different than, say, getting exposed to the antigen of a virus in a vaccine and then developing antibodies to that. This is simply a priming response. Very, very important, relatively new and somewhat unknown concept. So here is a macrophage, the most prolific of the big eaters. That's what the name means. Macro big phage is eating. This macrophage is eating Candida albicans. In the past, I've joked, a newly discovered organism. Of course, it's not newly discovered. Candida is something we've known about for a long time. The million-dollar question when you see this picture is, what induces the macrophage to eat that Candida? How does it know that it's something that it should go after instead of ignore? Well, there's some kind of molecular structure on the surface of that Candida albicans, specific to yeast, but specific even more so to that kind of yeast that is moderately to severely inflammatory. That activates the macrophage to go into action and engulf the Candida. So it's all about what's on the cell surface of that stranger, a somewhat dangerous stranger, that alerts the macrophage to become activated. Macrophages were first discovered, they're really the first immune cells to be discovered. They were discovered by studying starfish larvae under microscope by Dr. Metchnikoff in 1884. The famous experiments he did where he stuck a thorn in the starfish larvae and he saw that thorn surrounded by cells. Well, those cells were macrophages. Now, these macrophages are now known to be a subset of the whole mononuclear phagocyte system. Almost all tissues have macrophages. The equivalent in the brain would be microglia, Kupffer cells in the liver. They look like amoebas, which makes you think that that's where they evolved from. They can move around like amoebas. And what do they eat? They eat microbes, they eat cancer cells, they eat debris. That's what makes them scavengers. They eat advanced glycation end products. They eat caramelized sugars. And like dendritic cells, they can stimulate adaptive immunity. Important to understand that macrophages are not always inflammatory. They can turn on the inflammatory process, but they can also turn it off. And I mentioned that earlier. We have the ability to turn off inflammation, and we do that as soon as we turn it on. If we didn't have this quick on-off, then every single inflammatory stimuli would produce kind of major reactions in the body, and that happens with people that have dysfunctional immune systems. So here's a picture of a macrophage getting activated. You see at the top right, a resting macrophage. There's some kind of inflammatory signal, perhaps exposure to a bacteria or a yeast. It starts to wake it up. It becomes responsive. So that macrophage is activated by the molecular structure. It starts to wake up. And then if it's immediately hit with exposure to pro-inflammatory cytokines, then it becomes fully activated. Well, once it becomes fully activated, it can go down a number of different pathways. It can become a dedicated phagocyte, where its main job is to simply clean up debris. It can become a cytokine producer, where its main job is to induce the proliferation of lymphocytes and differentiation of a B lymphocyte into a plasma cell. It can become a fully dedicated antiparasitic cell. So you see the molecular machinery inside of a macrophage is very complex and leads to all different kinds of possibilities. Let's switch gears now and talk about mast cells. And mast cells are something that a lot of my colleagues have been talking about recently. They're very, very interesting cells. Mast cells are basically the tissue equivalent of basophils. They're not exactly the same cell as basophils, but they functionally do a lot of the same things as basophils do. The difference is that mast cells are sessile. They're found in the mucosa connective tissue. They're found in the skin. They're embedded in the connective tissue of the skin. And mast cells are packed full of inflammatory mediators, not just histamine. Histamine is one of many. So they crank out all these mediators, they fill them up in granules, and then when they're activated, they either selectively release the mediators, they may just release histamine, or they may release the whole bag of tricks. It's really a fascinating study to try to figure out, how does the mast cell know exactly what's the mechanism involved that tells it to only release histamine or only release prostaglandin D2. How does it know? We don't really understand that, but we know that it is that specific. The mast cells really do sit on the fence between innate and adaptive immune cells. They are innate immune cells, but then they can acquire receptors made from IgE, which are clearly part of the adaptive immune system. What allows mast cells to degranulate? We see a picture of a mast cell on the right, full of all these granules, and all those granules are full of mediators. Well, they can be activated by direct injury. If you take your fingernail and you scratch hard on your forearm, you'll see a bit of a welt form, and that welt that's forming is a result of the mechanical stimulation of mast cells to release histamine and other mediators. Some people can just barely touch their skin and will get an urticaria, will get a hive. Those people are really histaminergic. They're really full of histamine and can react very easily. Sometimes not just a mechanical stimuli as well. The classic way that mast cells are activated is by IgE cross-linking by antigens, and you see that on the right. Let's say this is somebody who is allergic to penicillin. So they haven't had penicillin in many years, but their mast cells are still covered with IgE, and there's a very specific part of that IgE that reacts to penicillin. The penicillin will cross-link the IgE, which are really cell surface receptors, and that induces the mast cell to degranulate, which is what you see in the bottom right-hand corner. We also know that cytokines can activate mast cells, or complement split products can activate mast cells. Simply by getting a cut, we'll release complement split products, which will then bind to cell surface receptors on the macrophage and tell them to degranulate. As I mentioned, there are a lot of different mediators that are found in the secretory granules. Histamine is probably the best known. Another one is tryptase, which is a protease, but you also find serotonin, prostaglandin D2, various leukotrienes, proteoglycans like heparin, yes, heparin, platelet activating factor, PAF, chemotactic cytokines called chemokines. So there is really there are really a lot of different chemicals there, and they're mostly vasoactive. As you can imagine, they're causing vasodilation. They're one of the major players involved in what we recognize as the acute inflammatory process, the rubor, dolor, tumor, and calor. What's really new about mast cells, and the reason that so many people are interested in them, is the realization that they're involved in all kinds of diseases. Okay? It used to be thought that mast cells were mostly involved in allergy and asthma. Okay? They release histamine, they cause wheezing, they cause nasal congestion, etc. And so they were mostly thought to be players in the allergic process, and certainly they do that by increasing mucus secretion, causing vasodilation, edema, fluid secretion, and activating the inflammatory cascade. But now we know that mast cells are involved in all kinds of other diseases. Okay, the basic functions are pathogenic defense and wound healing. That's great. But these others are not necessarily a good thing. Allergic, anaphylactic reactions, autoimmune diseases, angiogenesis for tumors, obesity, mood disorders. Histamine is a neurotransmitter. So overactivation of mast cells can lead to overproduction of histamine, which then can affect brain function. Atherosclerosis seems to involve mast cells. Interstitial cystitis involves mast cells. This is a picture from Dr. Theoharides at Tufts University. He's really the master of mast cells. He calls himself the mast cell master. And he's attempted to map out how strong the associations are between mast cells and different diseases. The strongest association with asthma and atopic dermatitis, but also with interstitial cystitis, psoriasis,